This work is titled On the Spatio-Temporo-Rhythmic Mapping of the Task-Associated Brain Functional Networks by Haddad and Laleh Najafizadeh, Department of Electrical and Computer Engineering, Rutgers University. The outline of this presentation will be as follows. First, we define the dynamic functional networks. Then, we state the objective of our work, which is identifying the rhythmic, temporal, spatial, and task association characteristics of functional networks. Details about the rhythmic, temporal, spatial analyses and on extracting the task-associated functional subnetworks are given afterwards. Experimental data collection and results are given next, and finally, conclusions. First, dynamic functional networks. A functional network is a temporary structure spanning a set of brain regions which show high intersimilarities in their activity patterns during the corresponding interval. The similarity relations suggest a temporary collaboration among the involved brain regions on performing a common function. Since the underlying collaboration is temporary, functional networks are dynamic. Functional networks can be mapped in terms of their location in the brain, their dynamics, that is, their transitions as a function of time, and their rhythm, that is, the subband within which functional connectivity is established. Second, identifying the rhythmic, temporal, spatial, and task association characteristics of functional networks. Here, we propose a hierarchical framework for monitoring the multi-scale dynamic functional connectivity in the brain towards extracting the functional networks most associated with the task of interest. To best handle the multi-scale dynamics of brain activity, it is probed using electroencephalography or EEG. The degree of association of the identified functional networks with the task of interest is evaluated in terms of their recurrence rate during the recorded trials of the task. We start with the rhythmic analysis. EEG is decomposed using wavelet, then inverse wavelet transform into 6 dyadic subbands. These are the lower and higher parts of the delta subband, then the theta, alpha, beta, and gamma subbands. The subbands are normalized for subband equalization, then augmented into 6 band-specific sensor sets. Next is temporal analysis. For an interval of time, let our functional networks stay active, characterized by their common time courses or network activity patterns, and effective gain vectors, which represent the cortex to sensors conductivities. Note that each effective gain vector represents a spatial feature for the corresponding active functional network. Taking background cortical activity and channel noise into account, the sensor space EEG data can be formulated using the linear model. Therefore, a minimum mean square error estimate of the span of the effective gain vectors can be found through singular value decomposition, or SVD. Now, given the augmented EEG data at hand, the temporal analysis aims to break that data into intervals during which functional networks sustain their location and rhythm in the brain. Our previously proposed source-informed segmentation algorithm uses SVD to estimate the span of the effective gain vectors, which, given the augmented EEG data's nature, is now a spatio-rhythmic feature of active functional networks. Through that feature, our algorithm monitors the augmented EEG data for indications on changes in the spatio-rhythmic distribution of functional networks. That is, their distribution in space and over the different rhythms constituting the EEG signal. Spatial analysis. As we intend to locate functional networks on the brain, source localization is first applied to the augmented EEG segments to estimate the augmented cortical activity. Active functional networks are characterized by their common time courses, as in before, and now by their augmented intensity maps, which represent the participation levels of the nodes of the augmented cortex in each functional network. The augmented cortical activity can then be formulated in terms of the activities of these functional networks and other background activity. Then, a minimum mean square error orthogonal representation of the augmented intensity maps can be found through SVD. Now, the augmented intensity maps, which we just estimated for each one of the functional networks, are thresholded into Boolean maps. The thresholds we used are calculated via phase randomization-based Monte Carlo simulation. The Boolean maps indicate whether each node in the augmented cortex belongs to the network or not. Overall, a variable length sequence of segments per trial produces a corresponding sequence of functional networks. The last component of the proposed framework consists in extracting the task-associated functional subnetworks. Let A be a Boolean matrix whose columns represent the Boolean maps of functional networks that were identified during all the trials of the task of interest. The goal here is to extract a small subset of basis vectors that represent the most significant Boolean maps of functional subnetworks. Now, since each basis vector, and therefore each functional subnetwork, has its own corresponding occurrence pattern in matrix A or in the trials of the task of interest, the average recurrence rate of the functional subnetwork during the interval of the trial can be calculated from that occurrence pattern. This Boolean decomposition problem is solved here using the ASO algorithm, which provides an approximate solution to the problem. Having covered all parts of the proposed framework, we now move to experimental data collection. The data we use here is based on a visually cued motor execution imagery experiment, with tasks including left and right hand squeezing and tongue movement up and downward. The preprocessing applied to the collected data includes bandpass filtering and artifact removal using independent component analysis. Finally, the 3-second interval covering the planning and performance phases of each trial of the experiment are extracted and used for further analysis by the proposed framework. Results. The proposed framework is used to extract the 20 most significant functional subnetworks from each task of the experiment. Note also that the data collected from each subject is treated independently in our analysis. Below, we show the results for the tongue-up movement execution task for one subject. Each brain map below shows the location of a functional subnetwork. The color of the subnetwork gives the rhythm within which functional connectivity is established among the corresponding brain regions according to the color code. In this example, most of the extracted functional subnetworks formed within the higher part of the delta rhythm and the beta and gamma rhythms. Note also that most of the extracted functional subnetworks appeared in about 65% or more of the trials of the given task, as given by their trial recurrence rates. To summarize the example earlier, here we show one functional subnetwork from each one of the 6 rhythms. The maps of these functional subnetworks are shown to the right, whereas their dynamic recurrence rates are shown to the left using the same color code. The functional subnetwork in the gamma rhythm, in red, covers frontal, temporal, and motor regions of the brain, as can be seen from its map to the right. Whereas from its dynamic recurrence rate curve, also in red, one can deduce that this functional network only forms during the task execution phase, starting 1000 milliseconds after the beginning of the trial. Similarly, the functional subnetwork in the beta rhythm, in blue, covers prefrontal regions of the brain and have higher recurrence rate within the task execution phase. In contrast, the functional subnetwork in the higher part of the delta rhythm form within the visual cortex and have 2 peaks after each one of the 2 visual stimuli. Conclusions. Here, we proposed a novel computational framework that can probe the state of functional connectivity in the brain during the task of interest through multi-trial EEG recordings. The framework identified and mapped the extracted functional networks in terms of their rhythm in each one of the EEG subbands of interest, their dynamics during the trial, their location in the brain, and their association degree with the task of interest. The proposed framework showed promising results when tested using experimental EEG data. Thank you for watching.